RNA purification by organic method. There are different types of the RNA purification. By using those protocols, we can purify the RNA. The most common protocols or methods by which we can purify the RNA, that is RNA isolation by salt precipitation, total RNA extraction by organic method, affinity purification, and selective messenger RNA isolation. So these are some of the most common methods by which we can purify the RNA from the cells or from the tissues or from the body fluids. So here we will see the procedure how we purify RNA by using organic method. So in case of organic method, the first step is lyse or homogenize the cell by using triazole reagent or phenol chloroform. Because the name of this method is organic method, it means during this procedure or during this method we use the organic compounds. So in this case, usually triazole reagent or phenol chloroform, they are used to homogenize or to lyse the sample, and then the samples they are centrifuged. That is the first step of this protocol. During the second step, organic phase, because there is phenol chloroform or there is triazole reagent, due to the presence of that reagent, the organic phase separates from the aqueous phase. So organic solvents, they are at the bottom, while RNA is present in the upper phase, which is aqueous. So by the presence of organic compounds, there becomes two phases. One is aqueous phase, which is present on the upper side, and the second is organic phase, which is present at the bottom of the tube. So during the third step, aqueous phase is on the top and RNA is present in the aqueous phase. Once both the phases they separate from each other, then cellular debris and the genomic DNA, they are present in the form of film at the interface of the two solutions, because lower is the organic phase, upper is the aqueous phase. The genomic DNA is usually present at the interface, that is a small film between the two phases. So these are the first four steps while purifying the RNA by using organic method. At the next step, RNA is removed, which is present at the aqueous phase, at the upper phase, that is removed to a clean tube, the next tube in which that aqueous phase is removed. Then in the next step, precipitate RNA and wash with ethanol. So once the aqueous phase has been taken into another tube, then precipitation of the RNA is done with the help of ethanol or with the help of isopropanol. Mostly ethanol is used in case of RNA. So then it is suspended RNA in water for further molecular biology techniques or further study the RNA. So if we see that what are the advantages of this method, which is the organic method. In organic method, we say this method is compatible with varieties of the samples, different types of the tissues or different types of the cells. We can purify RNA from different tissues and cells. This process can process small amount of the tissue or cells, as well as large amount of the tissue or cells. So in both cases, if we have less amount or if we have large amount, we can use this method for the purification of the RNA. So this is an established protocol. Many labs, they use this method for the purification of the RNA, as well as this method is inexpensive. There are some disadvantages of this method, which is called as organic method of RNA purification. There are organic compounds which are used. They are smelly and they can be toxic. And RNA contain contaminants of the DNA. Because DNA is present at the interface, Sometime while removing the aqueous phase from one tube to another tube, DNA also comes with the aqueous phase. So there may be contamination of the DNA during this process.